Hello everyone, my name is Supakan. I'm currently studying about the plants in genus Permane, family Permaneaceae. So today I present to you the evolution of the mycoheterotrophic plant focusing on the plastic genome approach. This is a part of my course of seminar code in Plant Science 2. First, I will introduce you some details about the plastome of the mycoheterotrophic plant. With this study, drastic reduction of the plastome size in the mycoheterotrophic plant, Tisme tentaculata relative to that of its autotrophic relative Tachycardia by Lim and the team 2016. And here the picture. There are a lot going on the left side. It represents the plastome of Tachycardia, which is the autotrope, compared to the right side, with less colorful is of Tisme tentaculata, which represent the mycoheterotrophic plant. The color it indicate the content, the gene content within the plastome. So you can see that of the Tisme, they a lot fewer colors. So that means there are a lot fewer gene contents compared to the counterpart, autotrophic. Also, the size is a lot smaller for the Tisme. And why they still there, the plastome, why it remains? It's because the studies say that it's essential for the plant to keep some of that gene within the potted. It affects the survival of the plant, like shown in these yellow boxes that I highlighted. For the turnip M, which related to the synthesis of some protein, and the turnip that for the synthesis of the heme, and the ACCD, which is involved in the synthesis of the fatty acid, which is essential for the plant to live and survive. So that's why the parts still remain there and not totally disappear. Also, this study compare the mycoheterotrophic plant to the other heterotrophic plant. In this case, it's the parasitic plant. The example in this is the Raphacea lacaceae. Both have the reduction of its genome, the size, and the contents within it. And in the extreme cases, like in the Raphacea lacaceae, the part is totally lost. But somehow it's managed to store some information about the gene in the prostate, how to reconstruct the parties, because they found the structure that look like the plastic, but it's not in the flower tissue of this Raphacea. That means within the life cycle of this plant, the plastic-like structure can reappear. That means there's some data that kept somewhere else, not in the plastic. They thought that it might be that these genes contain this genetic data might migrate from the plastic back to its original in the nuclear, so it can be recalled later. And it ensures that this secondary function of the plastic is crucial for the plant to survive. For the next, I will talk about this study, the plastome on the age of the evolution breakdown of mycoheterotrophic plastic genome by Graham and team 2017. I pretty like this figure. It shows that these taxa have like wide range spectrum. I mean, for the mycoheterotrophy, they show the range from initially mycoheterotrophy, like in the orchid, that the seedling of the orchid need the fungi to germinate it and to thrive before it can survive. It can live by themselves, to the partially and the fully mycoheterotrophic. And this will show that this process is tend to be in one way, not the other. When the plant go in the like the route of being mycoheterotrophic, it tend to stay like that and not go back. For the initial load, it's thought to be that the gene that tend to lose first is the NADH, the hydrogenase-like genes, with related in the for the response for the oxidative, photooxidative, which is the state when the plant facing the sunlight, like too strong sunlight. Sorry, yeah. It involved in this process about the ferredoxin and ADP reductase. I mean, the gene is involved in this system. So the story is the plant that survive in the like understory habitat, it doesn't have to expose to a very, very strong sun. So this no longer necessary for the plant, this response. So why waste the result to do what you don't have to do? So eventually these genes get the load in the way of the evolution, and when this load, the plant cannot go back in the other habitat, like not under the canopy of other big plant in the tree, in the deep forest. So this plant have to invest more in mycoheterotrophy to survive, because it cannot go back to be in the strong sun anymore, which is what I said before, that this tends to be the one-way irreversible process. This chart clearly show how the pattern of the losses of the genes and the evolution of this mycoheterotrophic plant started. Yep, like I said, initially the NADH, these hydrogenase like genes start to lose first, and then when it cannot go back to its previous habitat with a lot of sunlight, it tend to lose the ability to photosynthesize due to the fact that it load the essential gene for the photosynthesis themselves, and then it lose all that related genes to the photosynthesis. But there are stop sign here that why the plastic remains, because in this case, this study said about the 5 core non-bioenergetic genes is about like plant making food genes. So like it's essential for the survival of the plant itself, but not related to the photosynthesis. So the plastic remains. And why? Why, when we already have the nuclear genome, why we have to study the plastic genome? It's because if you look into the plant cell, there only one nucleus, but there are a lot of plastid in comparison. So for the example, if you have to extract the DNA data to be analyzed for the phylogenetic, isn't it easier to do with the higher copy? Like when you have to use the herbarium specimen with the DNA pretty not in the intact form. You better have a lot of data for the analysis, for the comparison, and it conserve enough. Still, it diverse enough to in the intergeneric scale like that, not inter species, to compare in the phylogeny. But like in this case for the mycoheterotrophic is adequate to be analyzed, because in different plants is have the erosion rate differently. So that's why we still use this plastome, not just the nuclear genome, because they have its own pros and cons. So the last study I'm talking about will be the phylogenomic inference in the extremities, a case study with the mycoheterotrophic plastome by Lame and the team 2018. What is the challenge that's posed for study mycoheterotrophic plant using the plastic genome? Because in mycoheterotrophic plant, they evolve pretty quick. For the plastome, because it's not vital for the plant to conserve these sequences, because they don't do the photosynthesis anymore. So the gene in plastid itself, it can evolve quicker, because if it evolves not in a good way, still the plant still survive, because this is not a protein coding gene anyway. So this means that this can lead to the misinference of the phylogenetic position on the tree. I will show you why. In this case, this is the maximum likelihood tree. This is a part of the tree. You see these red long branches. These are the mycoheterotrophic plant that show the weed long branches. These distant on the tree, the length, it indicates that how different the data on the sequence compared to it adjacent clade. So this means that this compared to this, it's very, very different. And normally this, the long branches, it indicate that something might go wrong on RT. So this study they do the comparative tests for the method of this analysis, that is it actually wrong or not. So they compare the maximum parsimony and maximum likelihood and the raw DNA data and the amino acid sequence that translated from that DNA data. Which one should give a better result? The parsimony based is the simplest analysis for this. So what is wrong here? The previous long branches, it tend to go together because the algorithm of this analysis is compare how different or how similar each taxa or like compare together all together. And when a lot of taxa that have very different but similarly different, so it tends to group that long branches together. So they call this a long branched attractor, which when you look back on another tree in another study, this group in the wrong position. So the result wasn't congruent with other studies. The next is the maximum likelihood analysis. So this show the result that congruent with other study. I mean, like the long branches and the support are good. It sit in the same position as the previous study that didn't use the plastome for the study. And DNA versus the amino acid did show that the DNA gave a better result, because when you translate the DNA to the amino acid, it's not like one codon per one amino acid. So like these, like in this L amino acid, leucine, I guess, it can be translated from not only these three bases, it can use other codon to translate into this. So the data that we have is lost in the way when we translated it. So the support of the amino acid sequence when you do the analysis tend to be lower than the DNA data. So you better use the DNA data in this case. For the conclusion, use maximum likelihood with the DNA data for the better result. And here, before we end the overall conclusion of this study, the genome size of the mycoheterotrophic plant indeed decreased compared to the counterpart autotrophic. In the evolution, the gene contents are loads, but it's not like randomly loads. They got a pattern that how it start to load and where to stop. And at that stop point is the core gene that still remain, because it has the secondary function that like lethal for the plant to survive, to live on. And the evolutionary base using the plastome is more than just possible. You just have to use the right tool, like in this case the maximum likelihood with the DNA data. And you can study the evolution of this plant using the plastome genomic data. It's possible, which is good. And here the reference flag 2 demo, just a nice picture. Thank you very much, and that's it for the evolution of mycoheterotrophic plant. using the plastome. Thanks.